Hello, my name is Nick Plowman and I'm an oncologist at St Bartholomew's Hospital in London. And it gives me great pleasure to introduce my esteemed colleague and close friend, Chris Parris, with whom I've had the privilege of working for 25 years. Chris is an authority, an international authority on all aspects of DNA repair. Together, me as the very junior partner, we've described two new DNA repair syndromes. I've always found Chris to be extremely knowledgeable on all aspects of laboratory techniques, but most of it has been revolved around DNA repair. In the last few years, Chris has mentored a PhD student through a project on circulating tumor cells and their separation. And I can't think of a more ideal authority to lecture us this evening on the technology of harvesting methods. Chris, over to you.
Okay, thanks very much, Nick, and a very warm welcome to everybody to our webinar on circulating tumor cells. As Nick has said, my name is Chris Parris, and I'm a professor of biomedical science at a university in Cambridge in the UK called Anglia Ruskin University. This is a university of about 35,000 students, so it's actually somewhat larger than the other university in Cambridge that you may have heard of. And if I may be so bold, I'd like to think as good a university in some ways. But in any case, tonight I'd like to take you through a brief review of some of the main platforms for CTC isolation, harvesting, analysis, and possibly downstream processing. So what are these differences and why is it important? So what is the best platform? Is there a best platform for identifying circulating tumor cells? And how might we use the kind of data and the kind of analyses that we can derive from circulating tumor cells in these various different methods and platforms for isolation and identification? How can we use that information clinically and also from very much a fundamental and basic research point of view? So just to take you through the brief content of what I want to go through today, we'll provide a brief definition of circulating tumor cells for those colleagues that may not have studied this particular area, and then focus a little bit on the biological and clinical relevance of circulating tumor cells. And then we'll get into the main body of the talk where we'll look at different platforms, different methods that have been developed for identification, enumeration, and capture of CTCs. A very important development in CTC biology of late is the phenomenon of Ctc clusters, so groups of CTC cells that seem to break away from primary or secondary tumors into the circulation. And these might be very, very important in terms of driving metastasis and tumor development and tumor progression within an individual. I'd like to focus a little bit on what you can do with CTCs and clusters. How might we culture them? How might we perform further analysis to not only make clinical decisions, but to ask some very fundamental questions about tumor biology, about metastasis. And then at the end, we'll try and summarize and give some kind of state of play. And hopefully that will provoke a lively discussion when we complete our webinar. So CTCs, the basic fundamental fact is 90% of cancer deaths are caused by tumor metastases. And unfortunately, malignant tumors have the ability to shed cells from their leading edge and in a very well-defined process invade surrounding tissue and enter the lymphatic and the circulatory systems. And these cells that break away from the primary or tumor are known as circulating tumor cells. And they can potentially lead to metastatic lesions in distant sites within the body, some distance away from the primary tumor. So in terms of their biological and clinical relevance, generally CTCs are quite rare, considering they will be a very, very minute proportion of the other species of cells that will be in the blood, red blood cells and a wide variety of lymphocytes. Thus, their detection, counting, enumeration, and molecular characterization are very challenging. In order to interrogate the information that we might get from tumor cells, we need to have efficient and viable platforms for the separation of circulating tumor cells from a variety of other cellular species within the blood. But the attraction here is that if we can develop really good methods for CTC analysis, we have a minimally invasive tool to find out something really important about the clinical status of an individual with cancer, and a very minimally invasive way to derive important research material. So they serve as a really useful, what we call a liquid biopsy. And the general thinking is, and there's plenty of evidence out there, that CTCs mimic tumor properties. They conserve tumor heterogeneity, they drive metastasis, and circulating tumor cells in many ways retain many of the properties of the tumor from which they were derived from. But we might want to consider also that CTCs are a part of a portfolio of other important biomarkers that are important within cancer diagnosis, response to treatment, early diagnosis, prediction, and molecular phenotypes. And it's worth also just bearing in mind, we might return to this at the end of our webinar, that they're a part of a portfolio of useful biological material, including exosomes, tumor markers, tumor proteins, tumor-educated platelets, circulating tumor RNA, and circulating tumor DNA. And we might want to look at circulating tumor cells as a part of a portfolio of other important biomarkers that might tell us something very, very important from a clinical perspective and a research perspective about the nature and progression of an individual's cancer. Looking at a sort of brief historical timeline of CTC capture and evaluation, this is remarkable that we're not looking at a sort of a new phenomenon here. I was able to search through the literature and I found the first ever publication by Thomas Ashworth in 1869, where he saw in a post-mortem of an individual that had died of cancer, tumor-like cells within the bloodstream. And this was way back in 1869. Moving on to the 20th century, it was around about 1959 when CTCs were beginning to be isolated by filtration, and then bringing ourselves up to sort of around about the early 2000s, 2004, when one of the principal platforms for CTC identification, cell search, was approved by the Food and Drug Administration in the USA. So there have been platforms around for a good number of years, but I would say that the technology is moving in a very fast fashion and very innovative way to develop sort of gentle and methods that will preserve the integrity and structure of CTCs and potentially Ctc clusters for future analysis. So right up until about sort of 2014, kind of the emphasis around sort of circulating tumor cells was the identification and counting, and how different numbers of circulating tumor cells within the bloodstream of cancer patients might equate to the extent of that cancer, might equate to whether or not a patient might be responding to treatment. But as we move forward up until the present, there's been more of an emphasis on capture, culture, downstream processing, so capturing the circulating tumor cells, culturing them, analyzing them, determining if they can be used for other processes, such as learning fundamental sort of information about the processes of metastasis, and also whether or not we can use perhaps cultured and fully analyze circulating tumor cells to make important clinical decisions, such as patient stratification, for example. So some common perceptions around CTCs, that there are CTC capture and evaluation technologies being developed and commercialized on many different devices. I think one of the issues around CTC capture and evaluation and the various different platforms that we have is that they're very different in the way in which you handle and manage the cells, and very different in the way in which they capture and enumerate cells. So we are perhaps lacking a little bit of standardization across the field here. And I think, if I may be so bold, that this may have led to physicians and researchers, fundamental researchers, being uncertain as to the clinical, perhaps diagnostic and scientific value of CTC cells and the technologies that are there to isolate, enumerate and characterize them. But there is no doubt that this is a coming science. It is growing in validity, perhaps. It is growing in popularity, and there is a strong desire to be able to make important clinical decisions on the basis of circulating tumor cells. So moving into kind of methods for CTC evaluation and capture, I think we can probably break the processes of identification, evaluation, and enumeration of CTCs into two fundamental different types, which can then be further subdivided. So we can characterize one batch of CTC devices as antibody-based, immunoaffinity-based capture systems, and then secondly, size-based capture systems. And these approaches and these methods can be further subdivided to give an extra level of detail. So if we look at this flowchart here, we have sort of whole blood here. And if we look at immunocapture, we can use antibodies and magnetic techniques to capture cells of interest, rare cells of interest. And then we can select in a positive way markers that would bind to CTC cells and therefore pull CTC cells away from components of whole blood. Or we can go in the opposite direction, where we can target non-CTC cells, pull those away from our cancer CTC cells, and then be left with a population of cells that we want. So we can have immunomagnetic methods. And then there are microfluidic methods, which are a slightly different technology to immunomagnetic, but they're using kind of filter devices, stepwise exclusion devices that we can then further positively or negatively select the rare cells that we seek. So there's the immunocapture methods, and then there are size-based methods, which might be filtering cells through a membrane or through some kind of microfluidic device based upon 2D and 3D geometries, which we will focus on a little bit later in our webinar. And then there are simple density-based methods, and we've all probably carried these types of methods out in our experience, where we might use phycohypate and density centrifugation to separate out red blood cells from lymphocytes and circulating tumor cells. So it gives us sort of an overview of the different types of methods that we might consider in terms of CTC capture and enumeration. So just to then focus a little bit on enrichment methods for CTC capture, we can use positive enrichment methods where the device or magnetic surface is bound to antibodies that recognize a particular marker that might be expressed by our cancer cells. In this case, EpCAM, which, if you're not familiar with EpCAM, is an epithelial cell adhesion protein that regulates calcium-based interactions, cell-to-cell interactions between epithelial cells. So here we have a method for pulling away our CTCs while leaving intact or leaving within or allowing to flow through our device non-cancer cells with a pan CD45 lymphocyte marker. So we've got an immuno-enrichment method there, relying on a marker that identifies cancer cells to pull our rare cells away from the components of whole blood. Or you can go in the opposite direction, where you use an immunomagnetic surface with antibodies bound to pull away non-cancer cells, so that we're left with CTCs that are label-free for further downstream analysis. So in other words, technological platforms for capture and analysis of CTCs rely on biological properties of cells and physical properties of cells. So biological properties might, for example, as we've discussed before, the expression of cell surface proteins, cell adhesion molecules, the ability of cells to undergo invasion, perhaps whether the cells are viable or whether they've become apoptotic. So we might be able to exploit some of these biological properties for CTC identification and evaluation. Or we can exploit physical properties such as cell size, the density of cells, the deformability and compressibility of cells, or perhaps their electrical charge. And there are platforms and methods that focus on all of these different types of biological and physical properties that can be exploited. So, taking that categorization a little bit further for CTCs, for example, immunoaffinity, where we're looking at positive enrichment. So antibodies, as I said earlier, are targeting tumor-associated antigens linked to, for example, magnetic particles to capture the CTCs. And one of the principal methods here that we'll focus on very shortly is the cell search platform, which I think even to this date is the only platform for CTC identification that has received FDA approval for use in the clinical setting and clinical scenario. Or you can go in the opposite direction for immunoaffinity, which is negative enrichment, so antibodies targeting leukocyte-associated antigens linked to magnetic beads to deplete unwanted cells. So here we can exploit the human CD45 pan-lymphocytic marker to deplete those cells that we don't want, and we're left with those rare CTC cells. Looking at some of the biophysical methods, so we've touched on earlier density gradient centrifugation. So as I say, that many of us that have worked in this type of area may have done Buffy coat isolations using Feickel-Hypake type methods. Well, these are some of the density centrifugation methods that we can use with or without a porous membrane above the separation media that gives us a further level of separation of, for example, red blood cells, white blood cells, and potentially our precious circulating tumor cells. Or looking into biophysical methods, which I will focus on as we get a bit further into our talk, that focus on microfiltration type methods in 2 and 3 dimensions. So in other words, size space separation using pores and 3D geometries. So let's focus on one of the platforms that has been given FDA approval, and that's cell search. So it's an immunomagnetic type platform where CTCs based upon EpCAM expression in the first instance, or the cell adhesion molecule that I talked about earlier, is used to positively enrich for our circulating tumor cells. We can also use a number of other markers to perhaps validate and verify the existence of our circulating tumor cells, such as DAPI, which is a very common nuclear marker used for many immunological immunocytochemical type experiments. Also the epithelial marker cytokeratin, which is an important part of the cytoskeleton in many epithelial cells. And we can also give ourselves another level of kind of exclusion using CD45 antibodies, a marker to exclude white blood cells from our analyses. So just to give you a quick sort of talk through then in terms of the process that we might go through. So we might be left with, we might start off with a blood sample, for example, shown in the tube here. And then we have antibodies that recognize EpCAM in a ferrofluid linked to a magnetic surface. So as our antibodies that recognize EpCAM, they will hopefully capture the precious circulating tumor cells. And there may also be an immunomagnetic setup where we have a similar kind of capture device for circulating tumor cells, so leukocytes. So we can use this immunomagnetic separation technique to separate out our Ctcs from everything else. And this all occurs within a magnetic cartridge and a fluidic device that comprises the cell search system. So to give you a kind of example of the kind of images that you might derive from a cell search analysis, and this is from a typical example of the type of data that you would get. This one was published in Clinical Cancer Research by Yap et al. So we have a number of panels here. On the left, we have cells that are nucleated, as shown by the blue DAPI stain. And then we can see in red cytokeratin, so that is confirming that that is likely to be an epithelial derived circulating tumor cell. Then we have our pan-CD45 leukocyte marker. In this particular example, we've also got a marker for the androgen receptor, because we are, in this particular study, we were looking at prostate cancer cells, which is shown in green, and then a composite image there. And what we can see, which is quite useful for us, is that if we compare the DAPI with the cytokeratin stain, we can see that the cytokeratin is verifying the identity of at least in the upper panel one circulating tumor cell, but the smaller cell just below that is looking like it's a lymphocyte of origin, because that is being identified or labeled by the CD45 pan leukocyte marker. So it gives a really potentially very good way in which we can separate out our circulating tumor cells from non-circulating tumor cells, and provide a reasonably accurate enumeration of circulating tumor cells within a cancer patient. So what about clinical utility? Well, certainly FDA approval for breast cancer, at least. And there are many studies out there showing enumeration of Ctcs linked to metastatic progression, linked to overall survival, and also response to therapy. So some of the examples I've given there was an example in terms of overall survival, where in non-small cell lung cancer, there was an evaluation of the number of Ctcs within a defined unit of blood that might be associated with worse overall survival or a better overall survival. And the data that was sort of coming out of these type of studies and many other studies, that if you have patients with 5 or more Ctcs per 7.5 mils of blood, then their overall survival is significantly worse than patients with less than that critical number. And this is where we're getting into the concept of thresholds, because for many different cancers, there seems to be thresholds of circulating tumor cell levels within the blood that may be associated with a worse or better prognosis in terms of overall survival. So, and there are lots of studies out there for breast cancer, certainly for non-small cell lung cancer, that show that you can make these clinical correlations between the numbers of circulating tumor cells in the blood and overall survival. There are also many studies out there which demonstrate that a reduction in the number of Ctc cells in cancer during therapy is associated with a response and a tumor response, a tumor reduction. So again, some really nice evidence showing that Ctc levels in blood enumerated by cell search can in some way mimic overall or be linked to overall survival, and also linked to responses to therapy. And these are really kind of useful clinical utilities. So as I've said, cell search was cleared by the FDA for cancer diagnosis and cancer evaluation some 15 years ago for breast cancer. I think its potential and full clinical potential and utility has still to be fully demonstrated for some of the reasons that I've not previously identified, but I will talk about in a minute. And there are emerging large-scale studies which are trying to demonstrate predictive value, but there still needs to be a lot of work in this area. So taking the clinical utility aspect a bit further, can we use this type of data from cell search to perhaps evaluate for early clinical development of drugs? I think we need more data there. Can we use Ctc cells on the basis of cell search for patient stratification, for example, treatment with targeted therapies? I think these questions remain unanswered, but there is some emerging evidence which we'll talk about in a minute. But there are limitations to cell search and immunology-based methods in terms of sensitivity, specificity, and tumor heterogeneity. And we'll come to this in a minute. So in terms of patient stratification, a recent publication just a number of weeks ago from the Ctc randomized trial, which was basically sort of demonstrating that it might be possible to use Ctc cells to evaluate whether or not a patient should be given either chemotherapy or endocrine therapy as a first-line treatment in hormone receptor positive ERBB2 negative metastatic breast cancer. So here there is evidence emerging in clinical trials that suggests that Ctcs, on the basis of the type of technologies that we've talked about, may help in the decision-making process in terms of patient stratification for either standard chemotherapy or endocrine therapy, certainly within breast cancer. So some fairly positive data there. However, there are some fundamental limitations, and certainly what I want to talk about is the limitations in and around the use of markers such as EpCAM. So EpCAM is, I would say, a fairly kind of promiscuous marker, and also a marker that suffers from the fundamental concept of tumor heterogeneity. And this is a little bit of data that was derived from my own lab, using the novel technique of multispectral imaging flow cytometry. And what we were able to show when we were trying to enumerate circulating tumor cells, in this case from stage 4 ovarian cancers, that we were identifying cells that appeared to be circulating tumor cells, but were not expressing EpCAM. So if we look at our panel A here, we have a cell showing in bright field, as you would see as you look down a light microscope. Then on channel 2, which is our EpCAM staining, immunocytochemical method of EpCAM staining, showing beautiful EpCAM staining, and then we can identify our nucleus, and similar for panel C. But we also have these large epithelial type cells that are not expressing any EpCAM. And also we can demonstrate from this type of analysis, you get all kind of staining intensities in between. So there's clearly a proportion of circulating tumor cells that are reflecting the fundamental property of cancers, and that is tumor heterogeneity. So you do need other mechanisms to identify and to capture circulating tumor cells. And many other investigators will sort of do double staining techniques where they might use cytokeratin, for example, and they might try and separate these out from mesenchymal or non-tumor cells using a stain such as the intermediate type 3 filament marker Viamentin, which is a characteristic of mesenchymal cells. So there's the whole tumor heterogeneity issue that we have to deal with, and the heterogeneity in the expression of specific tumor-associated biomarkers that we might use for Ctc capture. And then there's the whole concept of epithelial to mesenchymal transition. And many cancer cells, as they progress towards, as they develop further, will lose some of their epithelial markers and adopt mesenchymal properties or non-carcinoma type properties. The conundrum here is that as our epithelial markers are lost and the cancer cells adopt this more mesenchymal type stem cell type properties, it's often associated with highly motile and invasive cancer cells, which might be very, very important in driving metastasis. And unfortunately, EMT can be associated with increasing invasiveness, chemoresistance to treatment, and of course, the metastatic phenotype. So if we have a proportion of our circulating tumor cells that have undergone EMT, or epithelial to mesenchymal transition, then using these epithelial type markers to identify circulating tumor cells might be a fundamental flaw in the way in which we want to identify and measure and evaluate circulating tumor cells. So cell search clearly has a very, very important and fundamental role in the whole circulating tumor cell biology and evaluation. But there are other systems out there that rely on different types of properties, physical properties, and filtration type properties for the identification of circulating tumor cells. And I'm going to focus on a couple of these other type methods. Certainly, I want to talk a little bit about the ISET system and also the Parsotix system. So analysis of CTCs using ISET. What is ISET? Well, it literally stands for the isolation by size of tumor cells. Reported some 20 years ago in the American Journal of Pathology by Professor Patricia Patoloni-Braccio. And ISET relies on the use of specifically designed filters to allow the elimination and selection of our precious circulating tumor cells away from most of the leukocytes within the sample. So I apologize for the complexity of this slide, but essentially what we have here in panel A is our blood collection from a patient that's mixed with a buffer. Then we apply this to our ISET filtration device, and essentially what the filtration device is a membrane that has these small pores of a defined size, such that smaller lymphocyte cells should be able to pass through these pores, whereas our circulating tumor cells are trapped because they're just too big to squeeze through these holes. So if you consider that the average normal eukaryotic cell is perhaps 6 to 8 to 10 micrometers in diameter, if we have filter pore sizes that are in excess of these sizes that represent perhaps our larger circulating tumor cells, we can hopefully trap them and then recover these cells, or actually stain them in situ, perform an enumeration and an evaluation of what we have there. So we can use whole blood and conduct a filtration. We can separate out components of the blood and conduct a filtration. Or we can conduct a filtration and then downstream processing that by perhaps magnetic methods and further depletion methods, such for example antibodies bound to CD45, to further refine our separation techniques. So looking at sort of a typical filter separation that ISET might use, it brings into question some really important sort of fluid dynamics. And this is all about sort of retaining the integrity and size and shape of our circulating tumor cells, and indeed the viability of our circulating tumor cells. So here we look at the top view of our filter mechanism, and here are our pores, and here's a cell trapped over one of those pores. Hopefully our smaller lymphocytes are passing through and the red blood cells are passing through. But then we have the theoretical situation where we have a potential circulating tumor cell sitting over one of these pores, but we still have the fluid dynamics rushing past this particular tumor cell, which may increase the flow rate or the fluid dynamics or the pressure or the pascals, if you like, unit of weight or the unit of pressure against a specific area that might be rushing past this cell. So this might increase the PSI either side of the cell, but cause a reduction in PSI right over the cell itself. And this may cause all kinds of compressions, all kinds of relaxations, but in other words, it's subjecting that cell to shear stress. and this may affect the viability of that cells, and it might actually destroy some of those cells. So we might actually underestimate the number of cells that we have present within our blood sample. So we've got the potential, while these are useful mechanisms, there is this slight potential where we can lead to individual cell damage, as well as disruption of clusters, Ctc clusters, which I want to come to in a minute. And you can see the potential changes in, or at least potentially visualize the changes in pressure as we move either side of the cell and over and around our captured circulating tumor cell sitting over our pore. And you can actually see the theoretical distortion of the cell as it's trying to squeeze through the pore. But this sort of gives an idea of the type of filter separations that you can perform. So looking at a comparison of Ctcs between cell search and ISET, there's a rather nice publication that looks at a direct comparison of cell search and ISET, published a good number of years ago by Farace in the British Journal of Cancer. And it compares cell search against a number of histological different types of cancer, including breast, prostate, and lung cancer. And I apologize for the complexity of this slide, but I don't really need you to look at all of the information. We have a number, about 20 patients with their age, histology, and this is for metastatic breast cancer. We've got the hormonal status shown in this column and the TNM classification. And what we've got is a comparison of circulating tumor cells in 7.5 mils of blood between our ISET filtration system and our magnetic capture system, cell search. And you can see that the numbers do not always correlate. There are some examples, certainly for breast cancer, where you identify fewer Ctc cells with our filtration device than you do for our magnetic capture devices. There's very substantial differences between in our patient B3. And you also have the corollary of this, where you might identify more Ctc cells with ISET compared to cell search. But I think it very sort of fundamentally shows some of the differences in the way in which Ctcs can be identified and captured and evaluated based upon your different techniques that you're using to capture your circulating tumor cells. So this is sort of for breast cancer. Moving on to look at prostate cancer, again with ISET and cell search. Again, we've largely metastatic prostate cancer shown here. Given the PSA evaluation here, and again, Ctc cells in 7.5 mils of blood under ISET and also cell search. Again, we see this separation in the number of cells that we identify, differences between ISET and cell search. So on the one hand, we've got a patient number 3 where we see 248 cells under ISET, Ctc cells, and 90 in cell search. But you can see some very, very huge differences between the two platforms in the number of cells that are being identified between the two methodologies amongst this group of 20 patients, and that's for prostate cancer. Looking at lung cancer, a similar sort of picture. Some correlations, so for example, in patient L7 for lung cancer, where we have a significantly high number of Ctcs identified with ISET. Apologies, this should be ISET next to the TNM stage. And over on the right here, we have the number of Ctcs identified using cell search. So certainly I get the message that I wanted to bring across here is how we do see differences depending upon the platform that you're using. And you can do your correlation coefficients, and as you can see from this slide, fairly low correlation between the number of Ctcs identified on these different platforms for these 3 different types of cancers. So sort of comparing cell search and ISET, there seems to be a lack of correlation, a lack of consistency in Ctc recovery between the 2 approaches, between an immunomagnetic and a filtration type device. And again, I will sort of qualify, this is a very limited evaluation of just some of the fundamental systems used for the identification and enumeration of CTCs. I would say there was, within this example, this useful paper, a lack of consistency in terms of CTC recovery and the pathological stage of the tumour for lung, breast and prostate. But it's a similar picture if you look at lots of other publications comparing CTC capture methods. So I want to move into looking at another system, but before we do that, I just want to bring into the discussion the concept and the phenomenon of Ctc clusters and how this might be a really important sort of development and discovery and a really important feature in future CTC biology, future CTC diagnostic and clinical perspectives. So looking at clusters, kind of the points that I want to make here is that most CTCs in the blood have a very, very short half-life. They don't last long, perhaps 2 to 3 hours. The circulatory system is quite a hostile environment for a CTC cell. They are subject to mechanical stress, environmental damage, shear forces, oxidative stress, immune attack, and potentially also apoptosis. And those CTC cells that are going to be able to drive metastasis must have some kind of survival advantages. And one of the ways in which circulating tumour cells might acquire that survival advantage is in the form of Ctc clusters, 2 or more Ctc cells that are released from the tumour. And certainly publications would suggest that when Ctc clusters exist in a fairly high level within the blood, you generally see a high level and more efficient metastatic process with poorer clinical outcomes. And while their formation is yet to be fully understood, they're likely to be released, of course, from the leading edge of the tumour, contain highly motile and enzymatically active tumour cells, have this survival advantage and drive metastasis. And here we can see on the left-hand side of the slide what we mean by clusters. So we've got a number of cells here, some of which are displaying our EpCAM epithelial marker. Others are displaying the cytokeratin marker. These cells might also be displaying epithelial to mesenchymal transition because they are expressing the viamentin mesenchymal marker. So we have a very, very complex and heterogeneous collection of circulating tumour cells in this particular cluster. So I think the discovery and the biological relevance of clusters is becoming very, very important in the whole CTC biology. So in terms of the identification and isolation of clusters, it presents particular challenges. They are particularly rare. Perhaps one cluster per 10 to the 7 leukocytes and 10 to the 10 red blood cells. They also have the potential to dissociate during capture and processing. So it's very important that we develop kind of gentle methods with gentle fluid dynamics to maintain clusters within their native form. And of course, these clusters vary in their physical, cellular and molecular characteristics, as we saw from that immunocytochemical image in the previous slide shown here. There are many platforms, as I say, developed for the capture of CTCs, but only a few have shown potential for the isolation of intact clusters. And here, I think it's important that we look at this kind of fluid dynamic sort of microfluidic device that is called the Parsotix CTC capture device, devised by Engel PLC. So looking at, and this is a device that is designed to reduce shear stress and pressure on cells as we go through the isolation process. So it relies on a sort of a microscope cassette, if you will, that has multiple channels within that cassette, allowing flow of our sample in multiple directions. But the important thing is, within the cross-section of our slide, we have this stepwise size differentiation as our sample, our blood sample, moves through the cassette. So you could see that within this cassette, we could capture a large group of clusters in this particular area of the microfluidic device, whereas smaller cells are going to flow right through, and when we might actually capture single circulating tumor cells within this stepwise smaller region shown here. So it's a single-use Ctc separation cassette. The sample containing our rare cells, our Ctcs and clusters in whole blood, is routed through the cassette under a constant pressure of about 5 mils per hour. So it's a slow but steady isolation process. The cassette structure is based upon step configuration from perhaps 4.5 to 10 micrometers. So it gives us the opportunity to capture circulating tumor cells and clusters of different sizes. So looking at this in different dimensions, what we've looked in the previous slide, we've looked at the side view, and we can see this stepwise gradation as we reduce the size in which the cells can pass through. But it's not the only channel that's allowing the passage of fluid. So while we might block this particular channels, we have multiple channels of fluid dynamics passing either side of the cell. So we are retaining a steady PSI within the region of our circulating tumor cell and our cluster. So it has the potential to maintain cellular structure. It reduces potentially the shear force and stress that we are applying to our rare cell, the cell that we want to capture because we want to do some biology or some clinical diagnosis on the basis of that. And we're maintaining a steady pressure. So it's very important that we consider just not the one dimension of separation, but also the fact that this is occurring over a large kind of number of channels, over a large volume, and over a large space of up to 600,000 micrometers wide. So it gives us this opportunity to maintain the structure of our cluster or our circulating tumor cell. And similarly, if we look at clusters, here is our cluster that's being captured in our stepwise cassette, our Parsotix cassette, but we're still maintaining flow either side of the cluster. So we are maintaining steady pressure. We're not increasing or decreasing pressure behind the cluster or the circulating tumor cell. So we've got a chance of retaining the full structure of the cluster there. So coming back to sort of looking at CTC isolation in cell search and Parsotix, again looking at some prostate cancer patients. Again, this is one example of a paper that was published a few years back by Xu et al. In PLOS One. And we can see that in terms of a number of prostate cancer patients, the number of circulating tumor cells that were retained and captured seems to be significantly higher than what we've seen with cell search. So this rather more sort of gentle mechanism of fluid dynamics that we have running through our Parsotix system might give us an ability to retain many more circulating tumor cells, and perhaps give us a more accurate reflection of the number of circulating tumor cells within an individual patient. And we can also see the identification of these clusters here shown, DAPI in blue on the left of this immunocytochemistry panel, and then cytokeratin, a good marker of epithelial cells, the pan lymphocytic marker CD45, and there is our merged image. So we've looked at a magnetic device, we've looked at a filter device, we've looked at a microfluidic device, and they all have slightly different methodologies and features. But I think the next step that we need to look at is what we want to do with CTC clusters as CTCs and clusters subsequently to the identification, capture, and enumeration. But I think, for example, the ability to do something biologically or research-wise or clinically with our CTCs post-isolation might provide a really useful new class of biospecimen. So I'm going to suggest possibly that a biobank for circulating tumour cells might be a really important thing that might answer some very fundamental questions around CTC biology. The ability to identify CTCs, and particularly clusters, might create new tools to investigate the cascade of molecular events leading to metastases. High throughput platforms, as we are developing with Parsotix, for example, might be useful for screening therapies that arrest the process of metastases and provide critical advance in the precision management of cancer by allowing proteomic, genomic analysis and also an evaluation of patient stratification for targeted therapies. But I think it's really important that we can capture cells that are viable and retain many of their active tumour properties. In terms of just touching on a couple of methods that evaluate the viability of circulating tumour cells, I just want to briefly touch on the EpiSpot assay, which, whatever method you're using for the capture of your CTCs, you can check if they're viable due to the expression of something called S100 protein. Now, these proteins are a family of calcium-binding cytosolic proteins that have a broad range of intracellular functions, such as regulating calcium balance, apoptosis, migration, proliferation, and so on. So if your cell is expressing this protein, it is likely to be viable. Similarly, there are other viable CTC enrichment methods, such as the VITA assay, in which the ability of your cells to bind to a cell adhesion molecule scaffold, if they can bind to CAMs, the fluorescently labeled CAMs, there is a chance that those cells are going to be viable and therefore useful scientific and clinical tools to evaluate CTC biology post-isolation, post-separation. So to try and bring this to some kind of summary, the portfolio of CTC capture and analysis platforms is complex. Published data gives us really valuable insights into the use of these different platforms, but there are differences based upon methodologies used, based upon capture methods, and based upon individual experimental parameters described in publication. So we don't really have this standardization of methodology across different platforms. And I think there is perhaps still some reticence. I wouldn't necessarily, I've said confusion there, but perhaps that's a too strong a word. But, you know, some greater understanding required is what can we use CTCs for? Can we use them to answer fundamental questions about the biology of metastasis and the biology of cancer progression? Are they going to be useful in CTCs for cancer diagnosis and management? And I think that standardization is some of the areas that we need to focus on and perhaps have a discussion about very shortly. CTC clusters seem to be growing in importance and are likely to be very important drivers of the metastatic process. And validation and capture technologies for clusters, I think, is essential going forward. But I would like to make the point, and this comes back to a point that I made very early in our webinar, that CTC analysis perhaps should not be viewed in isolation, but as a part of a portfolio of other useful biomarkers for cancer, such as tumour protein markers, and as I say, circulating tumour DNA and RNA, exosomes, and tumour-educated platelets. And this may ultimately lead to the early diagnosis, prognosis and prediction, disease monitoring, and all the other things that we require and desire from circulating tumour cells. But just, you know, another kind of plea, if you will, about standardization. You know, we need to be able to have standard processes, standard methods, standardization of equipment. And one idea that I would like to sort of focus on is whether or not we could create a biobank of CTCs. So we capture viable CTCs, we can propagate them, we can make these available to the wider scientific community that will allow us to answer important biological and clinical questions. But ultimately, we would hope that this would lead to patient benefit. And that's the bottom line. We want to be able to use these to make critical clinical decisions that will lead to ultimate patient benefit and better survival. So I will stop there. I will thank you for your kind attention to our webinar. I'm really grateful that you've joined us here today. We're going to be happy to take questions. And thanks to Angle for sponsoring our webinar here tonight. So I'll now come out of presentation mode and we'll go back into panel discussion mode now. So thank you very much.
Thank
Thank you very much indeed, Chris. I'm going to hand over to Nathan Nagel, who will introduce the questions.
Could I first request that your webcam is on, Dr. Plowman, please? There we are. There just may have been a small delay. Okay, then. So we've got the first question from the audience is, so you mentioned earlier, Professor Paris, about the clusters and shear stress involved in some of the methods. Well, so how do the clusters and shear stress link? And in terms of what's the ramifications of breaking up a cluster, and how might that be a problem, you know, for the patient?
Well, I think if we accept the premise that clusters, the number of clusters or a critical number of clusters within the blood might be the important drivers of the metastatic process. If we use a capture method that might subject those clusters to excessive stress, we could break up the number of clusters that we have, and then possibly measure a higher number of individual circulating tumor cells rather than the clusters themselves. Because it would seem that the clusters are really going to be very important in the drivers of metastasis. Also, if we're subjecting those clusters to shear stress and we're breaking them up, what else are we doing to the cells? Are we killing the cells off because of the shear stress that we might be exposing them to? So I think it's whether or not we accept the kind of some of the emerging evidence that clusters are kind of really critical in driving the metastatic process much more efficiently than perhaps a single circulating tumor cell or a tumor cell or a single entity circulating tumor cell.
So just to follow on from that question, within the different methodologies, you know, which ones would you say are the top two that may be suitable to mitigate these issues that we've just discussed with regards to breaking up? And may I add as well that if these clusters are broken up, we don't know how that's changing the whole epigenetics of it as well, because then if you say that it may expose individual CTCs to more different environments plus the immune system. So if it was previously in a cluster and then it's opened up to an individual, it has changed the environment again.
Well, yeah, sort of coming back to one of the publications, and the title of one of the publications of that was "It Takes a Village to Metastasize." So if you've got, you know, it's safety in numbers, as it were. Sort of protection of circulating tumor cells within that cluster, that cells perhaps within the center of the cluster might be protected from some of the shear stresses, oxidative damage, potential DNA damage as well, and the cells that are within the center of the cluster might be even more viable and retain metastatic properties. Now, in terms of saying what are the top two, it's difficult to say, and I'm going to make myself sort of hostage to fortune. Certainly, some of the microfluidic devices that allow the circulating tumor cells or clusters to retain their shape and maintain PSI and maintain sort of a steady level of pressure that kind of mimic intra-capillary type pressures must be favorable, because you're mimicking what's going on in the body. So perhaps microfluidic devices that are not based upon kind of filtration, which subject cells to pressure changes, to shear stress, might in the long run be useful and more sort of valuable techniques for the identification of clusters going forward. But I mean, it's still an emerging technology, and there are still lots of, there's still work to be done here. But it seems to be an important innovative development going forward.
It's a primata, and I think that you partly mentioned earlier that I think it was Parsotix that was a gentle methodology. So
slightly gentler methodology, yeah.
Yeah,
and I think I think the Parsotix system is sort of coming into its own as we learn more about clusters, because we got the opportunity to retain the integrity of those clusters.
And then, as it's a part two of all this is the collection and then the analysis, and then and I suppose the next question is to Dr. Plowman. So just bringing this one down is, say, once the cells have been captured, so what next? Can we analyze the clusters? Can we analyze the subsets of the Ctcs? So if we talk about the ideal methodology, I know we're moving on to capture them, but what next? So Dr. Plowman, once the system has captured them, what next?
Yes, very good, and I'll get there slowly. A few comments on what Chris was just saying is that there's a paradox here. We think these circulating cancer cell clusters are derived from the leading edge and a consequence of EMT, but the fact that they are clustered together and suggests that the adhesion molecules, cadherin or whatever, which is thought to be lost in EMT, has to be retained to a certain extent for them to maintain their clustering. Whether there's some crosstalk between the cells as they leave the solid tumor as tumor buds is conjecture at the moment. But there's no doubt that cells can change their phenotype as they leave the leading edge through the blood to the soil where they migrate to. And in last week's oncogene, there's a good picture of some neuroendocrine cells that are avidly somatostatin receptor expressing as they leave the tumor, in the primary tumor, and yet when they reach the liver to metastasize, they've lost that expression. So there's clearly a change in the transcriptome, the phenotype, after they've left the primary tumor, and that's something that can certainly be probed in the future with CTCs. What can Ctc clusters tell us at the moment? Well, it can help you find the primary, because there's a couple of interesting papers now suggesting that it can help you tell where the tumor has metastasized from. The genomics of it will be complementary to the cell-free nucleic acid analysis, and I think that will be good. Yes, you're right. I think the current zeitgeist is that these cells do represent the phenotype of the tumor that is going to establish metastases. So that gives us a subset of the primary tumor that we can analyze that should show its aggressive features. And yes, we need to get down to next generation sequencing of that, and it's complementary to the other milieu in the blood, circulating nucleic acids, the mRNAs, and other features. Because in the tumor, just like the tumor microenvironment is important in the solid tumor, the blood is also adding to the phenotype, if you will, of those Ctc clusters. Those are my initial thoughts, and it's certainly going to be a burgeoning and important field.
And if I can just come in there and follow up on that, Nick, I think it's a really interesting question to ask: are there different species of clusters? So are there different? Is the primary tumor deriving different species? Are there metastatic lesions which are then are releasing clusters? So it'd be really interesting to know if there are different species of clusters, and indeed different species of circulating tumor cells based upon their genomic and proteomic analysis. And that might give us really important clues as to what is driving further tumor development within an individual.
Thank you both. A question to follow on is about ovarian cancer. So one of the questions that came and says, well, for ovarian, Dr. Plowman, are you with us?
I don't know what's happened to my vision at the moment, but here we go.
Okay, here we go. So in terms of different tumor types, is there various different CTC levels? And then in terms of looking at methodologies of collection of CTCs, there will be a whole wide spectrum where some tumor types produce a low. Is it the system's fault? Is it a different tumor type? You know, and the question came in about, is ovarian CTC collection low?
Well,
I mean,
it's a fascinating. Sorry, Chris.
You go, Nick. You go, Nick. That's fine. I mean,
Okay.
if I can just start off, certainly with the some of the data that we saw in the presentation, where we were comparing different methodologies of isolation with prostate, breast, and lung cancer, you were seeing very different numbers. So it would suggest that there is a methodology-specific input into the number of cells that we are recovering. Now, certainly, I've done a small study with a colleague at Mount Vernon Hospital in West London, certainly using stage 4 ovarian cancer patients, and we were seeing significantly high numbers of circulating tumour cells using imaging flow cytometry. But of course, that is a technique that's based mainly upon the identification of single cells. So again, I think it depends on the clinical and pathological stage of the tumour. There are different types of ovarian cancers which may behave in very different ways. I'm going to hand over to Nick to give some further insights into this, but I don't think there are necessarily some tumours that give really high numbers or low numbers. I think once a tumour is entering into the metastatic process, circulating tumour cells will be present. There are thresholds that it would seem that once you've reached a certain level that seem to drive metastasis in specific tumour types, but those thresholds might be different between different histological types of tumours.
Thank you.
Yes, I agree with that. Looking at the panels you put up for breast, there was a huge disparity, wasn't there, between the number of circulating tumour cells from different patients. Now, it will be interesting, and I'm sure that more studies will follow, where they will look at, say, triple negative breast cancer, which you might perceive to give rise to more circulating tumour cells than our luminal A, ER, estrogen receptor, very positive, no adverse features. Be interesting to see that. In your panel for prostate, P12 was of interest to me. It was a T1 N0 M0 prostate cancer, and you'd expect that to have a 90% cure rate by whatever methodology you used. I don't know the Gleason grade, but I mean, basically a T1 N0 M1 is a prostate tumor that's confined within the prostate capsule, is perceived to be curable, and yet there were circulating tumor cells. Isn't that fascinating?
Yeah,
and that must mean that there are others, and is it only in that 10% who don't do well, or does it give us a research project to see what else needs, what other attributes circulating tumor cells have for them to embed and develop as metastases? I found that a particularly interesting panel.
I think the point that Nick's made there is perhaps showing one of the advantages of circulating tumour cell kind of evaluation over and above some of the standard histological and imaging type techniques for cancer diagnosis, because there was a case where you would expect there to be, you know, if it's T1 N0 M0, you would expect there to be no release of circulating tumour cells, but clearly there were microscopic activities going on within that tumour environment that was causing the release of CTCs, even though it had been classified as non-metastatic, non-node involvement.
Thank you, Professor Paris. Question for Mr. Smith. So Mr. Smith is well read in this area, and then somewhere the papers have reported that there's a low CTC count. Could that be down to using some of the older methods where they've just, because of like limitations on EpCAM, so where they've said certain tumor types a low CTC count? Could it have just been that they used the older methods? And has there been any kind of papers comparing all the different methodologies to say, okay, then this is how this tumor type responds, you know, for the collection for CTCs? Could it be, because I know that there's different perceptions of clinicians and pharmaceutical companies to say, that's a low CTC count, or that's not where they're, but did they just use the EPCAM method, and have they looked at the new ones?
Yeah, no, that's a great question, and it's sort of the old thing of you see what you look for. I mean, there's a lot of very good data has come out of you know, over 15 years out of the use of cell search and EPCAM specific you know, capture techniques. Certainly there were a lot of puzzles in terms of some early trials. There wasn't an expectation that CTCs would, for instance, go up with metastases, and they didn't necessarily. Sometimes they went down, same with drugs, treatments. And as sort of the window is open a little more, and through a variety of technologies looking at sort of the full range of phenotypes, one can kind of go back and explain some of those studies by, you know, a decrease in epithelial, but there was a rise in mesenchymal type cells, and so the net went up. But because it was looked at in that narrow window. So I think, you know, that's a lesson. No technology, although obviously I will have my biases, sees everything. And I think it has to be approached from the basis of, you know, what is the question you're trying to answer? And clusters are a really good example of this, because I think the line was, it takes a village to metastasize. I think there's another paper which says never travel alone is how it starts. Because these clusters aren't just CTCs, you know, they could have neutrophils, platelet cloaks, monocytes. And as it turns out, that's very important. I think that, you know, there is some evidence out now that, you know, there's a big difference in the metastatic potential between a cluster of pure CTCs and one which has neutrophils associated. And this is likely a survival mechanism. Well, you can think if you used a negative depletion technique on that, well, it's been demonstrated you lose the clusters because of the CD45 cells associated with those. And so you have to look at every situation and understand what's the question you're looking for, and choose the appropriate technique. Clusters are really interesting. A comment earlier to me, because, you know, we as a device maker are really trying to do everything we can to retain these clusters. And the end goal of that is so that the biology can be understood. So in the patient, we can break up the clusters. And there are some trials along this way to reduce the metastatic. So it's a really interesting sort of comparison.
And then to build on that, Professor Paris earlier mentioned with regards to the lack of industry and scientific standardization of CTCs. I think it's an exceptionally good idea to look at the standardization and then, from a manufacturer, clinician, research point of view, that I've not read anything that's out there right now, and lots of different papers are saying, well, we do this, this, and this, and this. Yeah, but what benchmark? You know, what's your thoughts on a standardization so we can start to quantitatively analyze what is good, what isn't good, what are we measuring? Because how the hell do we agree otherwise?
Yeah, no, absolutely. And there clearly are a lot of very recent papers coming out comparing all the different techniques and illustrating these differences. So, you know, can't speak for everyone, but our approach has sort of been twofold in that regard. One is we've submitted our device, just the separation, to the FDA for consideration for approval for the isolation of cells. And the thought behind that is exactly that, to give a standard starting point. And we'll build assays on that. But that's not a trivial undertaking. So for that recent submission, we ran, just to do the analytical studies, ran over 15,000 blood samples. So it's not something you take on lightly. The second aspect, what we've heard from, you know, the people in the field is, again, the field's kind of moving from understanding sort of the principles of CTC isolation to more, what are they good for? You know, what can we tell from them? And in order to do that, you need standardization. So there's been a lot of requests, and we're moving forward, for instance, on standardized kits for mesenchymal, epithelial, et cetera, so that you can report the same answer, you know, in this lab versus another lab. And I think that's really going to help. And obviously that's been available from Cell Search and has been very helpful in being able to compare Cell Search study to Cell Search study, but being able to expand beyond that to the whole range of phenotypes, I think, is important. So.
And just if I can jump in there, Nathan, I think another important factor in all of this is you're going to put these devices hopefully into busy diagnostic laboratories in the hospital environment. So the whole question of training of operators, consistent use, the ability of the devices to be able to still perform and isolate circulating tumor cells and clusters from a blood sample that may not have been maintained in the most appropriate way, or one that happened to get that has been left on the bench overnight. That we still have devices that will compensate for the human element in all of this. So I think the training aspect, the human training, the operator training is going to be very, very important. So that, you know, if Fred does the test on Monday and then Jane does the test on Tuesday using similar buffers or even different buffers, that the test has been carried out accordingly, and therefore that we can compare between different operators and different days, different buffers, different batches. And this is all a part of the risk assessment that Paul has talked about, that we have to go through for validation of these types of devices.
Yeah, and
then the other device.
You've highlighted a very important point, and that's on the pre-analytical variables in terms of, you know, an assay. These are living cells, unless of course you put them in a preservation tube, and now you've changed their properties. And there's nothing wrong with that. Your assay needs to reflect the state of the cell. So, you know, it's no, I think we all learned that lesson on pre-analytical with mass spec, and it's no difference with CTCs.
So, thank you. Next question that's come in said, but in light of your general assessments, how about sensitivity is important in terms of cutoff? So generally cutoff is defined as 5 CTCs above 7.5 mil. What do you think of the sensitivity, and are some methods more sensitive than others? And is there a generally defined sensitivity which is acceptable? Who would like to take that question? Because I'm not quite sure who. Any volunteers on sensitivity?
I'm happy to make a couple of comments on it. And again, it kind of goes to how long is a piece of string. In a way, you need to define sensitivity. And one thing that's becoming clear is that enumeration alone, without phenotype, as we just talked about, is you know, maybe not the best biomarker information from CTCs. But then sensitivity is really, it tends to analytically be derived from cell lines. And this is a little bit of a unique challenge to CTCs. And cell lines are not the same as patient CTCs. And we just talked about how they can exist in different ways. They're very heterogeneous. And cell line to cell line, they will behave differently. And they're a good way of establishing reproducibility and sensitivity in terms of how many cells can you recover out of a spike sample for cell lines. But in reality, you really have to do patient samples, because these are, you know, so I guess my message is the real important part of sensitivity is the positivity rate in a particular disease state.
And you, Paul, you'll see an awful lot of publications out there which take exactly the approach that you've just said, where they will spike tumor cell lines into samples and measure sensitivity that way. And that, again, that doesn't necessarily tell us anything about viability or the biological and molecular and cellular properties of individual circulating tumour cells within different patients. So it's not an easy question to answer in terms of thresholds and sensitivities, because it will vary between each individual patient. But there are publications out there which seem to talk about thresholds, that seem to talk about certain numbers of cells are associated with greater or lesser overall survival and responses to therapy. But there isn't a standard line that you can draw. It will differ greatly on individual patients, individual histological types of tumours, and the individual biological and molecular properties of those circulating tumour cells. And let's face it, tumours are heterogeneous. Yeah. And that is a further complication in terms of trying to understand sensitivity.
Yeah, and a very exciting area that will partially address this, it seems, is progress in the ability to get, you talked about viable CTCs, to recover them and culture them, get them to propagate. There's more and more success recently, and those, if you will, patient-derived CTCs tend to behave much more like patient CTCs than traditional cell lines. And I think that will go a long way towards standardizing. There was a really interesting paper just 4 or 5 weeks ago that showed these cell lines derived from patient breast tumor actually could be identified, basically behave the same way when transferred into a mouse model as they progressed in the patient. And you could actually find markers early on that was going to lead to a brain metastases, typically. These are really exciting areas, and it takes, the ability to keep them alive for a while and hopefully propagate them in order to be able to do that. That opens up the possibility of drug testing and all sorts of other things.
I think this downstream analysis, downstream culture is kind of going to be really important going forward in terms of asking some of these fundamental biological research and clinical questions. I think once we can, the techniques are out there for the propagation and culture of cancer cells. There's a lot of experience already there, so this can be done. And I think, but you've got to have those methodologies in place that retain viable cells, and those are emerging.
Thank you. So questions coming with regards to building on the actual culturing side is, we've all read papers on the culturing of the CTCs, but does anyone know of any of the culture of the Ctc clusters? Has anyone read any papers on that? The question from Uday that's came in.
So there are some clustering cell lines available, and I think if particularly if you look at the, and I think most of them came out of Massachusetts General Hospital, but in breast cancer. But if you look at the papers, particularly of ACETO, there are clustering cell lines available.
Okay. Then in terms of probably following on from that, methodologies of analyzing it all, which are probably Dr. Plowman's expertise area. In terms of once they're looking at it, what are they looking at and why? I know, which is an very open question, but now you've got a cluster cultured. What would you do with it, Dr. Plowman?
The helping to define the primary is useful, because you may not know the primary, so that's one thing. And the next thing is, I think next generation sequencing on the tumor cells has to be of interest. It will tell us things as to whether the tumor has retained hormone sensitivity, or whether it's and whether you might even be able to do immunomarkers on it. So I think those are the analytics that we're interested in the solid tumor, but by doing it on the Ctc clusters, we hope to get an idea of the phenotype of the metastasis, the imminent incipient metastasis.
I mean, but if I can jump in there as well, Nathan. I mean, if you can downstream culture CTCs and clusters, then it opens up any area of study in terms of tumor biology. Now, from my perspective, going back to my roots and DNA repair, if I've got circulating tumor cells that are derived from clusters, I can ask very fundamental questions about the DNA repair capacity of those tumors. I can determine whether or not those patients might respond very well to more standardized chemotherapy and radiotherapy type treatment. So if we've got CTCs or clusters and that we are able to develop and we have the technologies for propagation of those cells, it opens up all kinds of analyses. It opens up the opportunity to do patient stratification. Are these cells expressing, can we stratify them for different types of smart or targeted therapies based upon their genomic expression profiles? So the ability to have this, really authentic material to do standard molecular, cellular, tumor biology, ask fundamental clinical questions, it gives us a new sort of species of biospecimen to start asking the very fundamental questions.
Yes,
and Professor Paris,
just one word is that 30 years ago, Sid Salmon started in Tucson culturing tumor cells, and the plating efficiency was so low that it didn't really represent the tumor. Now, hopefully, we've overcome that now, but it's just a word of caution. Yeah. You agree?
Professor Paris, building on the stratification part and then flipping over to Dr. Plowman, is if you can stratify the patients and identify, then surely it makes for a better clinical trial in pharma, because probably, I mean, it's been looked at, some of the patients that were on clinical trials where the drug failed, maybe they were just the wrong patients.
Yeah, and that evidence is emerging. We had an example there where CTCs might be able to drive the decision between standard chemotherapy or hormonal-based therapy. There are recent studies that have come out that you could potentially use CTC cells for checkpoint inhibitor type treatment for immunotherapy. So, those studies are emerging where you can potentially use circulating tumor cells for this patient stratification. Whether or not that falls within the bounds of acceptable clinical practice at the moment, that would be a question that perhaps Nick would need to respond to. But it could be a part of a portfolio of diagnostic information that we could have to determine patient stratification, to decide on treatment methods. I'll bring you in there, Nick.
I shared an interesting paper with Nathan the other day where people had actually taken not circulating, but malignant cells from a pleural effusion associated with a lung cancer. And they'd done various assays on them, including PD-L1. I mean, we know the cytology on them will give you the diagnosis of lung cancer. And they looked at the PD-L1 expression on these cells from malignant pleural effusion, and based their immunotherapy strategy on the result they'd got. So it's another example of, if you like, circulating tumor cells, this time in a malignant effusion.
And Dr. Plowman, in your experience looking at some of the clinical trials within pharma, where a drug has failed and people were disappointed, what are your thoughts on maybe they didn't select the right patients, or do you think that some good drugs in the past have not got to market, that maybe if they re-looked at them, just from a clinician's perspective?
Well, I'm not sure you could do a retrospective study without the CTCs, but certainly the phenotype of the CTCs would be relevant. If I was doing a study on CMS for metastatic colon cancer or triple negative breast cancer, I would love to be able to do it with CTCs and perhaps with the tumor as well. Somebody needs to do both and see which is most relevant, I guess.
I was thinking of the radiosensitizing drugs.
Well, you've got a world expert in Chris. Chris should handle that one.
So just repeat the question for me, Nathan.
It was looking
even international experts.
Some of the past trials have maybe not come to fruition because they didn't have the methodologies. I was thinking of radiosensitizing drugs. But maybe if we look at them now, it might be a different outcome.
Yeah, I think, as I say, it depends on now taking it further. I mean, it would seem that we have the devices now to retain viable circulating tumor cells, and I do think that opens up, to the opportunities to look at different drugs, to look at radiosensitive drugs, to look at things like PARP inhibitors, to look at the concepts of synthetic lethality in important, in those particular types of tumors, such as, BRCA negative breast and ovarian cancers and prostate cancers, such that we can potentially, examine radiosensitization. We can examine DNA repair in these samples, again, by isolating viable circulating tumor cells.
I can give you two good examples there. In ovarian cancer, there's a somatic BRCA mutation which comes along, and the way that this trial has been designed doesn't take cognizance of the fact that actually if you give them a platin and a PARP inhibitor, and there's a somatic mutation in BRCA, then actually you can push your carboplatin dose up, and it'll have a preferential effect on harming the tumor cells over the normal tissues, because the normal tissues, because it's somatic BRCA. And similarly, I posited the idea that, and as you may know, that HPV-driven cancers are more radiosensitive. Very good example is oropharyngeal cancer, where people are often reducing the doses of radiotherapy. Now, cervix cancer is driven by this, and if you have a differential effect due to HPV infection on the radiosensitivity of the tumor, and it's not in the normal tissues, then your chance of causing collateral damage is less. So you're quite right, Nathan. There are some radiosensitivity things that can evolve from that.
That can be answered.
It depends on the phenotype of the tumor. Sorry, Chris.
Yeah, no, I think again it's just reiterating the point. Give us viable circulating tumor cells, and it opens up the ability to answer many sort of fundamental clinical and research questions, not just for treatment, but as I said earlier, answering some of the fundamental questions that might drive the metastatic process. Evaluation of the expression of important matrix metalloproteases, looking at tissue inhibitors of matrix metalloproteases. So we can start asking fundamental questions about, cell-to-cell interactions if we have viable circulating tumor cells. So not just clinical questions, but important questions, scientific questions that folks like me want to answer as well.
Well,
Could I ask Paul?
It starts out with... Yes, Dr. Plowman?
Sorry, I interrupted. I apologize. Could I ask Paul how he sees circulating tumor cells interdigitate with cell-free DNA and serum factors, cytokines, and that sort of thing?
Yeah, absolutely. I strong, and I'm a protein person from way back when, so I'm going to go to serum biomarkers whenever I can. But I absolutely see them as complementary. There are, clearly there is a, if not clearly, I would say the evidence would point to CTCs representing, sort of an enriched fraction of, if you describe it, the leading edge, the most active part, maybe the metastatic status of a tumor. ctDNA certainly is much easier to obtain, and represents, in a good way, represents kind of an average population, much more than an individual CTC, which is very specific. So I think can provide two different kinds of information. The RNA is another thing that is a focus of our research, at least in the group here, because we see that as a way of profiling a CTC that should be able to give us some answers around aggressiveness and going back to the prostate cancer. So there's good evidence that CTCs, even in the pre-biopsy patients, might be able to predict what will be found as an aggressive form or a clinically insignificant form of cancer, when a biopsy is taking place. Exosomes are particularly in combination, because again, it's a different subset of biomarkers. And I think we need to look at all of these as not so much biomarkers on their own, but sources of biomarkers. The CTC can give you a phenotype and expression on the surface, the DNA, the RNA, enumeration, they're all different biomarkers. And, in ovarian cancer, we've looked very much at CTCs relative to some of the protein markers, and they behave differently in different subgroups. So one doesn't generally cover the entire spectrum of disease stage, et cetera. So absolutely complementary. The nice thing about all of them is, particularly in a time like COVID, is the ease of access for a biopsy and repeat testing, and it gives us the opportunity to look at something akin to PSA velocity, but changes over time in response to therapy, and that's changes over time, I think, are going to be very important in CTC relative to more so than absolute numbers, because they're so affected day to day by the status of the tumor and whatnot, but changes in, for instance, PD-L1 expression over time. So what percent of them are expressing PD-L1 in response to a therapy or not, I think is going to be a really key component.
Thank you, Paul. A two-part question. So first of all, starting out with Professor Paris and Mr. Smith, and then leading on to Dr. Plowman. So we've talked a lot about CTCs, but then earlier in the methodology, Professor Paris, you talked about in terms of the two-dimensional, three-dimensional flow of capturing the rear cell. Now, pretty sure that the method isn't just for CTCs, so therefore it can capture other rear cells. And as any oncologist would say, don't give me one set of biomarkers, I need more. So because one's not going to tell me anything. So Dr. Plowman, with looking at these systems in terms of capturing different cells, what else would you want to start making clinical decisions, ideally, as well as a CTC?
You caught me there. I've expressed an interest in seeing what if you weed out the CD45 cells, whether they're not an interesting cohort of cells to study in their own right, because we know that bone marrow derived cells not only are important in some tumors' growth, but influence it, and so that's a peripheral aspect that I've posited could be of value. I don't have any data to support it. What else would I find? I think it's what Paul said. I think it's the CTCs in conjunction with the serum factors, from cytokines to interleukins to non-coding RNA, in conjunction with them may have a big message on what's going on. I'd like to know Paul's critique of that statement.
Well, I mean, I totally agree. For me personally, and I'm not speaking for the company here, but I particularly find the potential in these associated CTC associated white blood cells, neutrophils, macrophages, etc., because they seem to be carrying let's just call it a cancer-associated profile, for lack of a better word. And I think that is, a way of, understanding maybe something beyond the tumor, maybe into the microenvironment, which is so critical for treatment. But there's other things that are turning out to be very interesting, and even outside of oncology, megakaryocytes is one that, have been identified, in Parsotix publications is associated with prostate cancer in a positive way. What's interesting is they, are starting to potentially show a role in COVID and severity of infection, and leading, maybe their role in leading to sensitized platelets. So that's another area. Circulating endothelial cells in COVID, but in cardiac disease, etc. I think this is no surprise, but this, again, if we can provide viable cells, it's a nice way to really start to probe the biology of those things outside of just circulating tumor cells. I think
also, if I can come in there, Paul, I think, if to really kind of establish the importance of circulating tumor cells in terms of clinical diagnosis and their utility is, will, for example, the number of clusters or number of circulating tumor cells, will that follow, for example, tumor markers, CA125 or PSA? Will the disappearance or emergence of circulating tumor cells mimic the reduction in the size of tumors as you go through treatment? So in other words, those really fundamental studies to track what's happening to CTCs and clusters vis-a-vis other standard diagnostic techniques. So, I guess a desirable outcome would be, will CTCs give us a single answer that would allow us to drive a lot of clinical decisions? Will it save, there's a health economics aspect to this. So will it make the diagnosis simpler? Will it make the process of arriving at an accurate clinical decision simpler and faster? And so those comparative studies, which are going on, are absolutely essential. But we need those comparative studies with the newer platforms, so that we're looking at clusters as well, not just CTCs. And when we're absolutely confident that the number of CTCs and clusters that we are recovering is a true representation of what's going on in a patient, then we can start answering those questions and start looking at CTC biology vis-a-vis other diagnostic techniques. And when we can match those, we can say, right, now we've got really powerful diagnostic method or patient stratification method that will make the whole process of cancer management much simpler. Now, I don't know if that's ever going to be possible, but that is surely a desirable endpoint to work towards.
Dr. Plowman?
So, Paul, I'll go first. Yes, very interesting subject. Now, that gets at the very topic as to whether numbers of circulating tumor cells can be used to screen for relapsing cancer. And it's far too soon, I think, to try and take Grail on at their own game looking at a screening test for cancer. But what you could look at would be for where you've done a mastectomy with curative intent to see if there's minimal residual disease, or to pick up very early relapse. And I don't think one analytic is going to be the answer, but circulating tumor cells would certainly be a favorite one analyte. Cell-free DNA, and in pancreatic cancer, you can look at ARAS or whatever. Grail have looked at the methylome. I'm not sure that that's what I would choose, and I think which analytics we choose is going to be a big subject for the future. But I would start looking at where you've done a cancer procedure with curative intent to see if you could pick up minimal residual disease or early relapse. That's what I would construct it, and I think circulating tumor cells could have an important role there, but not the only. I would suggest not the only analytic. Paul, sorry, I cut across you.
No, not at all, and I would totally agree with both those points. So going to what you just said, one of the, again, looking at protein serum markers versus a CTC in a recurrence. Very few protein serum markers are absolutely specific, but it's hard to argue if you're staring down a microscope at a tumor cell that there isn't cancer somewhere in that patient. So there are kind of different ways to look at it. But a very important point from Professor Paris, and that's also important commercially, is you know, none of these tests are any good unless you can get paid for them. And the only way nowadays you can get paid for a test is if you change clinical practice, if you give a piece of information. So prognosis now, unless it can guide a therapy, you know, you cannot get reimbursed for that. And the very first CTC test to get full reimbursement in the US is an example of that, and that's ARV7 on a CTC in prostate cancer. And you can make a very clear distinction of what drug to use at that point. And so that's, I think there's a lot of work going on in that area across many cancers, and it's really focused on, you know, can you provide information that will affect clinical practice? And that's what we have to dig out of the whole CTC and every other diagnostic test right now.
Thank you.
Yes.
Thank you, Paul. And Dr. Plowman, we talk about, you know, with patients, but at what stage do we start doing liquid biopsies on them in terms of super early or straight as possible? And, you know, and how many do they have to go through on this? And because then I'm just thinking from a patient perspective on, you know, and every oncologist that I speak to says, give me more data. So is it just one, you know, is it just one test, or can we use these samples for multiple tests? Just thinking from a patient perspective, and all the oncologists need much more data because obviously the complexity of it all. And it's just I don't think it's one company for any, just one decision.
At present, you can't use it early in the disease, although I've just given a very good reason why trials looking at it as being one of the analytics for seeing this early relapse is worthwhile, but that has to be proven in a level 1 way with a clear randomized trial. So we're using cell-free DNA and liquid biopsies, including CTCs at present, for patients with advanced disease, where we're looking at for clues to genomically targeted treatment that could help a patient, and that's what the cell-free diagnostic companies are. The Guardants, the Datars, these people are doing this, and I think circulating tumor cells fit in very well there. Once we can do a wide spectrum of next generation sequencing, you've mentioned ARV7, which is an RNA fusion, of course, and you can do that on the cells, but you can also look at other next generation sequencing or other gene expressions. We had a bile duct cancer the other day that was picked up as having a BRAF mutation, which was unexpected on if you like, a fishing expedition of genomics from a liquid biopsy. And I think now most thinking oncologists are doing fishing expeditions in advanced cases because they don't want to miss anything. And TRK analysis is a tissue agnostic mutation that is druggable, and we want to make sure we're not looking over a patient who could have that. I mean, for example, even secretory breast cancer can have a TRK mutation, and it's druggable. So I think that the next generation of oncologists are going to be doing a lot more of sequencing fishing expeditions and serially, because tumors evolve, as we know. I mean, cancer is a very good example of compressed evolution and obeying Darwinian, Lamarckian knock-on metabolome and Haeckel's theories of evolution are well exemplified as a tumor evolves. That's thought-provoking.
And in terms of the stage, so Professor Paris, do you see any of these methods being moved out into primary care or solely in the path lab at a cancer centre?
I think when I think of diagnostic tests, I think of them in sort of the hospital setting, in the high throughput environment. But you know, accepting Nick's point that with looking at individual patients, so that you don't miss anything with these kind of these detailed genomic analyses, provide some very useful insights and very useful options for alternative treatments. But I think, if you're going to use these kind of methods, they're going to have to go through not just FDA approval, but the equivalent approval in the UK. But again, it's what it's going to give you that you don't have already, what additional information that you can get. Getting any new diagnostic into the market is a very laborious process of, risk assessments, of approval, of conformity, European type approval. It's highly laborious. So, when I think of this type of thing, I think generally in, the path lab for maximum benefit of the maximum number of patients.
And then even on the usability,
oncological departments.
And then even on the usability part, because you mentioned about the different methods have still got human error in terms of how easy they are to use and how trainable, because ultimately as we scale up, there's only so many expert pathologists that can do it.
Well,
so
a part of the sort of a criterion of success is that these devices will work under and provide you accurate data under less than ideal circumstances.
Yeah.
So an inexperienced operator or an experienced operator will still be able to follow the protocols and deliver reliable results. That the devices will not be susceptible to changes in batches of solutions and buffers that you might need to put through the system. And all of these are part of validation processes. But there's a big issue of health economics. Does it boil down? Does it give you something new? Is there good value for money within the healthcare environment? And is it going to ultimately lead to patient benefit?
And I suppose that's subject to the stage that it's used and the results that it can give and the clinical change that can happen, which is, Dr. Plowman's department. Dr. Plowman, you said that you was currently using it in later stages. Do you see any place for the analysis early or even mid-phase?
I think let's start with the advanced stage and then look for and then move backwards. It needs to be established and accepted first. But then, yes, I mean, let's take an example. Let's take advanced melanoma. Okay, it's got BRAF mutation, and you drug it. You drug it with a single drug. Now, the escape pathways are not quite as random as you'd think in simple Darwinian terms. It tends to escape down the MEK pathway. And so there's level 1 evidence from a New England Journal paper showing that if you give not just one $100,000 a year drug, dabrafenib or encorafenib, that you add a MEK inhibitor, you improve the patient's not just interestingly disease-free survival, but overall survival. Well, so then you've got two $100,000 a year drugs that you're giving the patient on the expectation that you're going to block the commonest escape variant. Now, what it would be useful to do would be to have a liquid biopsy you could do to detect the early appearance of MEK upregulation. And if you had that, then it might actually be very cost effective to monitor the patient serially and wait for that escape mutation to arise. And that would be, you could argue, would save money rather than be costly. Does that answer the question? I can't remember what the question was.
I think it also answers the question in terms of health economics, is to say, the cost of oncology drugs. If you can make a difference and then decide whether to use it or not, it kind of pays for itself, really, because they're not £100,000 a test.
And I was going to make one other point. Yes, I remember now that, as somebody said earlier, liquid biopsies look at the mean of what's going on. They're not at the caprice of a single solid tumor biopsy. And so they do tell you the mean, and it may just be the most important mean because it's the leading edge, the tumor buds, the cells that are going to do it. But it isn't subject to the vicissitudes of a single site tissue biopsy of a solid tumor. it does look at the breadth.
Thank you, Dr. Plowman. So just final couple questions coming in now.
While you're looking at that, I'll just add that melanoma is particularly interesting with CTCs because there's some very specific markers for CTCs in melanoma. So back to the early question about sensitivity, that is a particular disease where they're easier to pick up earlier and well suited to monitoring, if you will. So.
Yes, wouldn't it be interesting to get together a trial with the BRAF inhibitor minus the MEK with serial CTC monitoring versus the two in combination? That would be a really nice trial.
On the basis of CTCs, yeah.
Yes, yes.
And it ties right back, I mean, the point about reimbursement, that's not just reimbursement for the diagnostic, it's insurance companies, and again, depends on where you are in the world, but whether it's nationalized healthcare like here in Canada or the UK or in the US with an insurance company, those are decisions that they you know, the administrators are looking for help for companion diagnostics.
Yeah, well, I'll give you another example, because in EGFR mutant lung cancer, we give, in England, we give erlotinib first line, but the FLORA trial shows that irrespective of whether the T790M is mutated or not, that actually the drug osimertinib, which covers both mutations, is better. But the license is not yet for that first line, and so there's scope there for looking at the time an escape variant comes up. It'd be another example of a trial you could do to see whether a liquid biopsy of not necessarily CTCs, it could be cell-free nucleic acids, would tell you when the escape variant is showing.
Thank you, Dr. Plowman. We've got a pharma question coming here in terms of, Dr. Plowman, you mentioned about trials, and a lot of pharma have used cell search in the past, and it's not always been fruition for them. So there could be some reluctance in terms of who drives the opinions to pharma. Is it oncologists that would drive it, or is it the research community? Who do you think who drives pharma if they've already been burned? So,
it's researchers like Chris Paris and Paul Smith who produce papers that show almost unequivocally that something is worthwhile doing, and they're accepted by then the ASCOs and the ESMOs and that sort of thing of this world, who then set standards. I mean, that's the way it goes. But it has to be based on research tests. You at the moment, I talk about doing fishing expeditions for advanced cancer patients. You can justify that, but for bringing it up front and earlier, you will need some validated studies.
But it's also not necessarily about pharma getting burned. What we're seeing here, and what is a fundamental thing, is that technology develops, technology improves, technology gets better, and as it should, because we have different ways of thinking about things. So I wouldn't necessarily call it pharma being burned. It's just there may be better systems that are developed as we understand more about the biology of CTC, the biology around their isolation, their survivability within the non-tumor milieu, that make us think differently around these things and gives us new opportunities. So I wouldn't necessarily say it's being burned, it's just being able to adapt to new technologies, new systems that allow us to answer different questions.
I think it's the risk aversion, isn't it, really, of like they don't know what they don't know, so they're not quite sure whether to spend it or not.
Yeah.
So
well, we can help farmers spend it, so that's no problem.
Yeah, that's fair. So we've got 5-10 minutes left now. So Paul, any questions? I'd like each of you to ask a question of someone else now. So because we've gone through a lot of audience questions. So Paul, you kickstart it off to ask a question of either Professor Paris or Dr. Plowman, please.
Wow. caught me off guard. I guess I'm, sort of thinking way out, and this question is for whoever would like to answer it, is again very intrigued by culture of cells, particularly if that can be done in a way that they, reflect where they were when they were captured over time. Is it as simplistic as, separating them out, testing a series of drugs independent of the research side? Where do you see that going, I guess, on the culture? What do you see that, if that's...
I think you can jump in there, Paul. I mean, again, if you're starting to culture cells in an artificial environment, there will always be some selection, and the more culture you do, as you know, you're going to get genetic drift of those cells as you go through serial cultures, and they may ultimately adopt a series of properties that are more conducive for the culture of those cells in vitro rather than in a patient. So it may be that there's this very small window over a limited culture period where the cells retain exactly the kind of diagnostic and cellular properties that you want. But again, that's based upon standard cell culture, cell line methods that we do know. And it remains unanswered as to whether that would be the same scenario with circulating tumor cells. I suspect it would, and that our opportunities to get the important scientific and clinical information is going to come very early on in that downstream culturing period.
So to me, as a developer, that translates to we need very sensitive methods on a very small number of cells. So, we can do those kind of tests immediately, if you will. Yeah. Thank you.
Dr. Plowman, what question would you like to ask?
It may be quite a naive one, but where you've got antibodies stuck to the cells, you need to knock them off, don't you, before without losing viability of the cells to do the testing. How do you get around that, and are there alternatives? How do you get around that problem?
Well, I mean, in terms of, you might use a proportion of your cells. So say, for example, you've isolated a nice big batch of circulating tumor cells through your Parsotix system. So a proportion of those cells might be used for your antibody type studies. But ultimately, you would retain a proportion of the cells that have never seen any kind of antibody so that you can do further analysis with them.
But you've got your antibodies stuck to them because you've used EPCAM or something antibodies to
So for example, if say for example I've used Parsotix and I've isolated 10,000 or 100,000 circulating tumor cells or clusters, I might take 10% of that of those cells and do antibody experiments with. I might do labeling experiments with, but so that's going to tell me something about the expression profile of different cell surface markers or intracellular markers. But I've still retained a lot of those circulating tumor cells that are still in their native form, and I haven't shown them antibodies, so I can still do other work with them, if you see what I mean. So it's about compartmentalizing your sample and using batches of it.
Yeah, and also it's, well, in the case of mRNA expression, where you're lysing the whole thing, it's about really good controls and very reproducible methods so that you can effectively subtract out the impact of contaminating cells. The antibodies in that particular case don't particularly interfere. Where they interfere the most is if you want to do live cell assays of any sort, then that is problematic. There are some approaches with cleavable linkers, etc., but again, every time you perturb a cell, you change it. So that's the point earlier.
And the people who use lysing techniques to look at differential apoptosis and lysis, what future there?
Well, they seem to be very attractive ways in which you can just lyse away certain cell populations and are left with, hopefully, a buffer that contains exactly the type of rare cells that you want to identify. But and they seem to be very good systems. I don't know what those lysing solutions may have on the full population of circulating tumor cells. So, and many of these lysing systems are proprietary buffers. So, but you may lose some cells, you may change the biological properties of some of the circulating tumor cells by using some of these lysing solutions.
Thank you.
I mean, a question that I'd like to come back to either Paul or Nick with is, you know, you find lots and lots of studies where they've looked at circulating tumor cell numbers in cancer patients, but there are very few out there where we've controlled that against the clinically normal individuals that don't have cancer, and there will be epithelial cells that will be in the blood system, you know, in the circulatory system of normal individuals. And I'm wondering, you know, what the data is around the differences of epithelial cells within normal individuals, and whether that is very different amongst different individuals.
That is a great question. And first point is you're absolutely right. And I guess I can assure you, when you run 15,000 healthy normal volunteer samples, you see the odd epithelial cell. And this goes back to very early in the discussion in your presentation when we talk about what's a CTC and the definition of that. And part of our efforts and other people has been as much focus on the positive identification as well as the addition of exclusion markers into a standard assay to deal with that. And when it's all said and done, you need a large population and you'll have a threshold. You know, once even with the combination of all of those markers. But you're absolutely right. And occasionally in a healthy normal, you'll see things that are definitely CTCs because you see them early on. But you're absolutely right, and that's really about systematic identification of what those cells are, and then testing the markers for them. So beyond 45, you know, panels of exclusion markers. So
thank you. It's interesting.
Thank you, Paul. And as keynote speaker, Professor Paris, you get to ask a different question. So it's for your next CTC study, what ideally would you like to do? Anything you will. What would you do?
You know, what's piqued my interest over, you know, the last few months, and certainly thinking about all of this technology, and it's just the importance of clusters, and really a good evaluation of or enumeration of linking the number of clusters within an individual to sort of metastatic potential. So it's really, I think, the significance of the role of clusters in tumor progression, the biological molecular properties of clusters. So I think, you know, it would be, for me, to be able to investigate the biology behind clusters and ultimately link that back to tumor progression, whether or not also those clusters could be used for, you know, making important clinical decisions such as patient stratification, and answering questions around the heterogeneity of clusters as well. Is it a single population of cells or a very, very diverse population of cells?
Thank you.